Hello, my dear students. Welcome you all to Sri Jayendra Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, in the previous lectures, we started with locomotion and movement. We discussed about the bones, the study of bones, that is osteology, study of joints, orthology, different types of joints, different types of bones. Today we are going to start with the muscular physiology. So, in this module, we will be studying about the structure of muscles, neuromuscular junction, contraction of skeletal muscle, that is sliding filament theory, and functions of skeletal muscle. The different types of muscles, when we talk about, especially for humans, there are three types of muscles. One is the skeletal muscle, known as striated muscles or voluntary muscles. Okay, they are attached to bones with the help of tendons. They are attached to bones with the help of tendons. The second category of muscles is cardiac muscles. We discuss about the cardiac muscles in the chapter body circulation and fluids. Cardiac muscles, they are structurally striated, is they have striations, they have bands, but functionally they are involuntary. And they are branched. They are present in heart wall. The third category is smooth muscles, which are unstriated. Or involuntary in function, and they are present in visceral organs. Present in visceral organs. So out of these three categories, we are starting with the skeletal muscles today. Skeletal muscles. The functions of skeletal muscles are as follows. They produce skeletal movement. They help to maintain body position. They help to support different soft tissues. They help in openings of guard, that is sphincters. They help to maintain body temperature. Very importantly, they help to maintain body temperature because of their contraction. And they help to store nutrient reserves. Okay, muscular glycogen. So these are the functions of skeletal muscles. They produce skeletal movement. They help to maintain body position. They help to support different soft tissues. They help in the opening of guards. That guards means the sphincters. They help to maintain body temperature with the help of their contraction, and they help to store nutrient reserves. Okay. So when we talk about the skeletal muscles, the starting, we will take the smallest unit of skeletal muscle, that is a myofibril. So if you will look at the myofibril, so this is a bone, okay, and the bone is attached to the muscle with the help of a tendon. Okay, this is a tendon. This is a tendinous joint tendon. Okay, these are the striations you can see. Now, the outermost covering, this covering is basically epimysium. Epimysium. Okay. So the muscle, the whole muscle is surrounded by epimysium. Okay. When we talk about one fasciculi, okay, one fasciculi, when fasciculi is the unit of muscular contraction. Okay. Fasciculi, there are numerous fasciculi like this. So we take one fasciculi. Fasciculi is guarded by perimysium. And when we take the muscular cell, okay, or a fiber or a myofibril, so this is the second fasciculi. This is the first. The outermost covering is epimysium, then comes perimysium, outside fasciculi, and the third is a myofibril or a muscle cell. It is surrounded by endomysium. So these are the three layers. Okay. So the structure of skeletal muscle is given. The skeletal muscles, they have striations, the bands. There are two types of bands when we talk about the A band and the I band. You can see the nucleus, it is multinucleated. Okay. There are no branchings. So all these properties are, I'm talking about the skeletal muscles. Okay. This is the muscular fiber or a cell. Okay. And these are the striations. In a pictograph, it will, under microscopic section, electron microscope. Okay. Scanning electron microscope, it will look like this. Okay. These are the cardiac muscles. Cardiac muscles, you can see the branches. Okay. Although they have striations, they have a intercalated disc, okay, and a branchings. Smooth muscles, they don't have any striations. Okay. So we will talk about the skeletal muscles in this module first. These are the muscle fibers I'm making. Okay. So these are muscle fibers. These muscle fibers are known as what? Myofibrils. Myofibrils. Okay. They are filled in between with cytoplasm, that is known as what? Sarcoplasm. Sarcoplasm. Okay. And the membrane is sarcolemma. This is the nucleus. Sarcosome, they are the mitochondria. And sarcoplasmic reticulum, it is equivalent to endoplasmic reticulum. Smooth endoplasmic reticulum. Plasmic reticulum. Okay. So muscle cells, they have myoglobin. Myoglobin is more affinited towards oxygen. Myoglobin is a specific skeletal and cardiac muscular tissue. Myoglobin binds to oxygen. Okay. When tissues are damaged, they release myoglobin. When muscular tissues are damaged, so they release the myoglobin. You can see the myoglobin, it is so affinitive towards oxygen that it is it is eagerly to carry the oxygen molecules. Okay. You can see in comparison to hemoglobin, myoglobin is more saturated to oxygen, more saturation to oxygen. Okay. The percentage saturation of myoglobin is more in comparison to hemoglobin towards oxygen. Okay. This is the three-dimensional molecular structure, quaternary or tertiary molecular structure of a myoglobin, and this is for hemoglobin. Okay. Skeletal muscle structures. These are the skeletal muscle structures. Epimysium, which is the outermost layer. These are the muscular fascillae. These are the muscular fasciculi. Okay, mean the muscular fasciculi is it is basically a bundle of muscle fibers. Okay, each muscle fiber is basically guarded by endomysium, and fasciculi is guarded by perimysium. Okay, so first comes epimysium, which surrounds the muscle. Fasciculi are surrounded by perimysium, and myofibrils are surrounded by endomysium. Okay, if you take a transverse section, okay, you can see the endomysium. The endomysium is surrounded the muscular fiber. Okay, so here you can see these are the capillaries. The outer membrane is endomysium. These are the myosatellite cells. Okay, and this is the nucleus. Okay, these are myofibrils, the axons of the nerves. Okay, this is sarcoplasm, sarcolemma, mitochondria. Okay, these are the fasciculi. Fasciculi. These are all of fasciculi. They are guarded by perimysium, and one muscular fiber is guarded by endomysium. Okay. Now, a muscle cell, it forms myofibril, which is basically made up of protein filaments. These protein filaments are of two categories: thick filament and thin filament. You can see the striations, nuclei. This is one myofibril. Okay, the sarcolemma is outside. Okay, this is the sarcolemma. These are the myofibrils. These are the myofibrils. So here you can see the thin filaments and the thick filaments. Okay, the internal structure is like this. The sarcoplasmic reticulum is here. Okay, then we have tubules. Yeah, this is the, it is having a striations. You can see the striations. These are the striations which are, you can see the striations. So these striations are A band and I band. A band is anisotropic and I band is isotropic. Okay. Anisotropic band, it is having substances of different refractive index. It is made up of substances with a different refractive index. Okay. So this is the A band or an isotropic band. It is between two Z lines or sarcomere. It is between two Z lines or sarcomere. You can see in the next slide. Yeah. So this is the A band or anisotropic band. It is between two Z line. One is here and another one is here. So between two Z lines, you can see the A band is here. Okay. Okay. So the A band is in between two Z lines. This is the smallest contractile unit, which is known as what? Sarcomere. Contractile unit. Sarcomere. Okay. So the A bands and the I band. Okay. Now, when we talk about the thick filaments and the thin filaments. Okay. So the thick filaments, you can see, contains thick filaments and these are thin filaments. So the thick filament, you can see here that they are having spokes like this. So these are thick filaments and these are thin filaments. These are thin filaments. Okay. Myosin and actin, they are the contractile proteins which are present in these thick filaments and thin filaments. Okay. So first of all, you can see the A band and the I band. A band is anisotropic band and I band is isotropic band. Okay. I band or isotropic band, it is made up of thin filament. In thin filaments are present and it is light in color. Light in color. While the A band or anisotropic band, it is basically made up of thick filaments. The thick filaments, along with the overlapping of thin filaments, okay, it appears darker under microscope. So sarcomere is the fundamental unit of muscular contraction. Fundamental unit of muscular contraction. Having the A band and the I band. A band anisotropic band between two Z lines of sarcomere. Okay, and the I band or isotropic band, it is the thin filament which is light in color. Understood? Each thick filament is surrounded by thin filaments and each thin filament surrounded by thick filaments. Each thick filament is surrounded by six thin filaments. Okay, and each thin filament is surrounded by three thick filaments. So I hope this is you get this, the thick filaments and the thin filaments. Okay, is that understood? Yeah. Now this is the anisotropic band. Okay, this is the I band or isotropic band. So the Z line basically divide the I band into half region. That is known as what? It is I by 2. This is I by 2. So basically between two Z lines is the sarcomere. So sarcomere is basically equivalent to I by 2 plus A plus I by 2. This is the sarcomere. Okay. So the A band or anisotropic band known as dark band. And the I band is known as what? Light band. The light band is having a dark line. This dark line, this is the dark line you can see, known as Zushinshibbi line or Z line. And the A band is having a light zone known as Henson's zone, known as Henson's zone. Just in the middle of Henson's zone, there is a present of disk, which is known as what? M line, which is known as what? M line. Okay. So the dark band is the A band having a light zone known as Henson's zone. Okay. And the light band is basically having a dark line known as Z line or Zushinshibbi line. Okay. The Henson zone is having a thick dark plate or disk known as M line. Is that clear? Yeah. Now, myofibrils, they are the site of contraction. Okay, and how the contraction it basically happens, you can see the A band and isotropic band. Okay, and this is the Z disc. Okay, this is the Z disc. This is the A band. So A band plus I by 2, it makes a sarcomere. Now you can see the H zone is basically, this is the M line. Okay, or Henson zone. This is the Henson zone. Okay. Now you can see there are two types of proteins, muscular proteins. These proteins are myosin protein. Myosin protein makes up thick filament. Okay, makes up thick filament. So myosin protein makes up thick filament. You can see the structure of myosin molecule. The myosin molecule is basically having a light meromyosin tail and a heavy meromyosin head, which makes the cross arm. Okay. So this is heavy meromyosin head and this is light meromyosin tail. So LMM, light meromyosin, and this is heavy meromyosin HMM. This is head and this is tail. Okay. The myosin filaments held together by M line protein and attached to Z line by the different protein. Okay. So here I will write the myosin filaments are held together by M-line proteins. Okay, they are held together by M-line protein and they are attached to Z-line by and attached to Z-line by titin protein. Understood? Now, you can see the heavy meromyosin head, it is having a cross linkages. Okay, you can see here spokes. Okay, these are the spokes. Okay, this is thin filament which makes, this is the thin filament. Okay, these are thin filaments and these are thick filaments. So these thick filaments slide over the thin filaments, okay, to make the contraction possible. Okay, now the myosin protein molecule is a hexamer basically the myosin protein molecule is a hexamer. Okay, it is having two heavy chains and four light chains. So myosin protein, yeah, these are the titin which I am talking about. Titin basically helps to make it attach to the Z line. Is hexamer. It is having two heavy chains and four light chains. Okay, myosin head is globular and shows ATPase activity. The head of the myosin is globular and show ATPase activity. Okay, so the myosin, the tail part is basically attached to the M line. I mean, the myosin, they are attached with the M line, and myosin to Z line is attached with the titin. Titin is a structural protein which is non-contractible. Okay, and it is the largest size protein in the body. Structural, non-contractile protein. Contractile protein. And it is also the largest size protein in body. So I hope this is understood. Okay. Now, this is the thick myofilament. Okay. The myosin molecule, the light meromyosin, LMM. And this is heavy meromyosin. This is the actin binding site. Okay. Here is the actin binding site. And this is the ATPase site. Okay. The two myosin molecules bound at their tail ends, okay, with the help of M-line proteins. These are the cross bridges, okay. This is the Bayer zone, which is not having the cross bridges. So we have discussed the thick filament or the myofilament, okay. This is the thick filament myosin, and this is the thin filament actin. So actin basically makes up thin filament, and the myosin makes the thick filament. These are the cross bridges, Z line, okay. This is titin protein, okay. So now, when we look about the actin protein, the actin protein is basically having also, it is also having, different other proteins which make the actin protein, okay. And it looks like a globular arrangement, okay, of small globules known as what, G actin. So G actin is a globular protein. G actin, it is a globular protein. Okay, just give me a minute. I'll add one slide. Yeah, this is the thin myofilament I was actually talking about. Yeah. So this is the G actin molecules, which is a globular protein. Okay. So these G actin molecules, they get polymerized. They get polymerized to form F actin. To form F actin. This is the F actin, which is a linear arrangement. And when these actin undergoes helical twisting, double helical actin strands, when they twist over each other, okay, so they make a highly convoluted structure, which is the thin filament. Okay. So here we have different types of proteins. You can see the tropomyosin. Tropomyosin is, I mean, there are two chains of tropomyosin. You can see. I will shade it with it red. These are the tropomyosins. Okay. So tropomyosin. It is a fibrous protein. It covers the actin sites during relaxed state. Covers the during relaxed state. Okay, so the tropomyosin is a fibrous protein which covers the actin sites during relaxed state. The other one is troponin or globular protein. So other one is troponin. It's the globular protein. It is having three units. The troponin is having three units. That is why it is known as what? Troponin complex. It is what? Troponin complex. So the troponin is having three units. Okay. TNT, or also coded as TPT, attached to tropomyosin. It is attached to tropomyosin. Tropomyosin. Another one is TNI, which is an inhibitor of actin. So it inhibits actin. And then comes TNC, which is a calcium binding unit. So these three units make troponin complex. It makes a troponin complex. So TN is troponin. TN stands for troponin. Okay. TN stands for troponin. Understood? So F-actin is basically linear. It is having two chains which are fibrous. Okay, and tropomyosin, it is having further two chains. Okay, which is fibrous protein. Okay, which covers the actin site during relaxed state, and troponin is a globular protein which is having three units. So the G-actin globular, it forms the F-actin. F-actin is two chains. You can see the twisting of two chains F-actin. They are fibrous. Okay, so tropomyosin is fibrous along with, they are also two chains. The F-actin is also linear, having two chains of fibrous, and the third one is troponin, which is globular, having three units. Okay, now there is a sarcotubular system which is not, present in smooth muscles. This sarcotubular system is not present in smooth muscles. Sarcoplasmic reticulum act as a calcium reservoir. Okay, so this system known as a sarcotubular system is not present in smooth muscles. It is not present in smooth muscles. Sarcoplasmic reticulum, it acts as a calcium reservoir. They have sarcoplasmic reticulum. This you can see the troponin and tropomyosin actions. Okay, the tropomyosin fibrous protein, troponin complex made up of three units: TNC, calcium binding unit; TNI inhibits actin; and TNT or TPT, tropomyosin, which attached to the tropomyosin. Okay. So this is the relaxed state. The moment calcium ions bind it, it becomes contracted. This is the contracted state. The calcium bound to troponin. Okay. The myosin binding sites expose. The myosin binding sites, they become exposed, and the troponin sites, they are attached to the calcium. Okay. The sarcoplasmic reticulum act as a calcium reservoir. Okay. Now there is a myoneural junction, which is the junction of sarcolemma and motor neuron. Okay. There is a presence of myoneural junction, which is a junction of. Okay. The sarcotubular system consists of sarcoplasmic reticulum and T tubules. Very important. Okay. In the previous slide, I'll write down system. It basically consists of sarcoplasmic reticulum and T tubules. Okay. Now, now we will talk about the neuromuscular junction, or we can say the myoneural junction. Okay. So here you can see the neuromuscular junction in which this is the myelin sheath, the axon of a motor neuron. Okay. The generation of action potential basically makes the nerves, to reach to its spike potential or the depolarization of nerve. Okay. So which basically releases the calcium ions, which basically releases the calcium ions. Okay. The proteins which are negatively charged, they've been attracted to the calcium which is released by cisternae of sarcoplasmic reticulum. Okay. So the proteins which are negatively charged, okay, they are being attracted by the calcium ions of cisternae of sarcoplasmic reticulum. Now you can see there is a release of neurotransmitter, okay, from these synaptic vesicles. These are the synaptic vesicles. They release neurotransmitter. These neurotransmitters, when they go, you can see the plasma membrane of the muscle fiber, acetylcholine receptor site, acetylcholinesterase, which break the acetylcholine. Okay, neurotransmitter gated channels. These are the motor end plates. The movement neurotransmitter, it comes in contact with these proteins along with T tubules, means sarcoplasmic reticulum, along with T tubules, that is sarcotubular system. Okay, the calcium ions is released. Invaginations of sarcolemma at the junction of invagination sarcolemma at the A band and I band junction in skeletal muscles. Okay, so these are the T tubules. They carry the T tubules, and then yeah, the skeletal muscle contraction. Okay, so the movement neuromuscular release, the release of acetylcholine, the axon terminal of somatic motor neuron releases the neurotransmitter. The somatic motor neuron releases acetylcholine at neuromuscular junction. Okay, net entry of sodium ions through acetylcholine receptors channel initiates a muscle action potential. The moment what happens, okay, as the acetylcholine is released, okay, the acetylcholine goes to the sodium gated voltage gated channels for sodium, and they open these voltage gated channels for sodium. Okay, and they starts the muscular action potential. Okay, so the sodium ions come in, potassium ions goes out. Okay, this is the T tubule. The T tubule is the invagination of sarcolemma. Invagination of sarcolemma at AI band junction. In skeletal muscle, the T tubules are found at the junction of AI band, skeletal muscles, while in cardiac muscles it is present at the Z line. These invaginations of sarcolemma at Z line in cardiac muscles. So the release of acetylcholine from the motor neuron endings on the somatic motor neuron axon terminal. Then the sarcolemma is depolarized. This is the depolarization of sarcolemma. Depolarization of sarcolemma. Okay, so generation of action potential. This is the motor end plate. Okay, the action potential is generated. Okay, now after this, what is going to happen? The action potential passes through the T tubules. You can see the passage of action potential through T tubules. The action potential passes through T tubules. Okay, so I'll write here. The third step is the action potential in T tubule alters the conformation of DHP receptors. Okay, so the action potential passes through T tubules and it alters the conformation of DHP receptors. Now the calcium channels on sarcoplasmic reticulum open and hence there is an exit of calcium ions from sarcoplasmic reticulum to sarcoplasm. So these DHP receptors opens the calcium release channels in sarcoplasmic reticulum and calcium enters the cytoplasm or sarcoplasm. Okay, understood? The calcium ions binds to the troponin, allowing strong actin-myosin bonding. Okay, so this is the third step in which you can see the DHP receptors. Okay, they basically open up the calcium release channels in sarcoplasmic reticulum and calcium ions enters the cytoplasm. The calcium binds to troponin, allowing strong actin-myosin bonding. Okay, the myosin head execute power stroke. This is the power stroke. Okay, the myosin thick filament. Okay, it execute the power stroke. Okay, the sixth point, power stroke. The actin filament slides towards center of the sarcomere. The actin filament slides towards center of the sarcomere. So this is the movement of actin filament towards the center of the sarcomere. Okay. Power stroke is the movement of myosin cross bridges pushes actin toward the center of sarcomere. End of the power stroke, myosin releases the bound actin and grabs another actin molecule. The cycle repeats. Which drives this process? The which process or which drive, what are the driving factors? Okay. So the junction of actin myosin bond is basically having is controlled by ATPase activity. Now, very important. The calcium, which is released by sarcoplasms, it binds to the troponin. Troponin undergoes conformational change and tropomyosin is shifted. Okay. So the driving factors is first is the ADPase activity. The second is the troponin undergoes conformational change and tropomyosin is shifted. And tropomyosin is shifted. Actin sites become free to bind with myosin. Because of the change in the conformation of troponin C, that is, which binds to the calcium binding site, which is having calcium binding site, the tropomyosin is shifted and the actin sites become free. After the shifting of tropomyosin, the actin sites become free to bind with myosin. To bind with myosin. Okay, is it understood? The molecular basis of contraction. Okay, the relaxed state, myosin head which is cocked. Okay, you can see the attachment of adenosine diphosphate along with the inorganic phosphate. The ADP plus inorganic phosphate forms ATP. Okay, The myosin has low affinity when ATP is attached to it. Myosin has low affinity when ATP is attached to it. Okay. The ACHE means the acetylcholinesterase. Okay. ACHE inhibitors lead to the continued contraction of muscles since acetylcholine won't be degraded. So there is a condition in which the ACHE with acetylcholinesterase inhibitors, they lead to continued contraction because acetylcholine is not broken down, it is not regenerated. Okay. The myosin has a low affinity for actin. Okay. Because of this low affinity of actin, it needs to enhance its activity. Okay. So the myosin bundle, when they attach with the myosin bundles, when they are attached, or the myosin head when it attaches with the ATP, okay, the myosin head shows ATPase activity, okay. So the myosin ADP inorganic phosphate is having high affinity for actin. The myosin has low affinity when ATP is attached to it, but it is having high affinity for actin when ATP is degraded to ADP and inorganic phosphate. Okay. So the cross bridges is formed when the ATP is broken down into ADP and inorganic phosphate. This is the myosin head. Okay. These are the actin binding sites. These are the myosin binding sites. The tropomyosin blocks binding site of actin. In relaxed state, tropomyosin blocks the binding site on actin. Okay, this is troponin. Okay, and this is G actin. Understood? So the ADP along with inorganic phosphate, it comes outside, okay, and the contraction happens. Only the head of myosin molecule move, not the whole myosin. Only head of the myosin molecule moves, not the whole myosin. Okay. The first is the cytosolic release of calcium. The calcium levels increase in cytoplasm or the sarcoplasmic, sarcoplasm, sorry. Okay, initiation of contraction. The calcium levels increases in cytosol. Calcium binds to troponin. Okay, TNC. Troponin calcium complex pulls the tropomyosin away from actin. Okay, so the troponin C, it basically pulls tropomyosin away from actin, and actin becomes free to bind with myosin binding site. Tropomyosin shifts exposed binding site on actin. The myosin binds to actin and completes the power stroke. It completes the power stroke. Actin filaments moves. The actin filaments moves towards the center. Okay, this you can see this is the release of inorganic phosphate from ADP. Contraction sequence of sliding filament theory. Very important. It was given by Huxley and Huxley. Thin filaments slide over thick filaments. According to this, the thin actin filaments, slide over thick filaments. Right? Sarcomere, because of the contraction, decrease in the length, the I bend decreases, H zone decreases. The main effect of sarcomere is on I band and H zone, sorry. I band decreases in length, H zone decreases in length. Okay. A band and M line remains unchanged. Remains unchanged. Okay. The acetylcholine, it basically helps to produce in the presence of acetylcholinesterase enzyme, which is a degrader of acetylcholine to form acetic acid, and choline, which is reabsorbed by synaptic knobs, synaptic vesicles, to again make acetylcholine. Okay. DFP, diisopropyl fluorophosphate. Okay. It is a nerve gas. It is an insecticide which inhibits acetylcholinesterase. Very important. So ACHE, acetylcholinesterase inhibitors. DFP, diisopropyl fluorophosphate. It is a nerve gas insecticide which inhibits acetylcholinesterase. Now you can see in the sliding filament theory, there is a tight binding in the Riger strait. Okay, the cross bridge is at an angle of 45 degree relative to the filaments, the myosin binding sites, the ATP binding site. It is at an angle of 45 degree. At an angle of 45 degree, the ATP binds to its binding site. Okay, on myosin, the ATP binds to its binding site on myosin. Okay, myosin then dissociates from actin. The moment myosin dissociates from actin, here you can see the picture. First, there was a tight binding in the Riger state, contracted state. The cross bridge is at angle of 45 degree. ATP binds to its binding site. The ATP binds to its binding site of the myosin. The myosin then dissociates from actin. Now, the ATPase activity of myosin hydrolyzes the ATP into ADP and inorganic phosphate remain bound to myosin. Okay. ADP, inorganic phosphate remain bound to myosin. The myosin head swings over and binds weakly to a new actin molecule. It comes forward, okay, and it binds to the new actin molecule. The cross ridge is now at a 90-degree relative to the filaments. Now, previously it was 45-degree, now it is 90 degrees, okay. If the calcium is present, then only it is going to happen, okay. The release of inorganic phosphate initiates the power stroke. The myosin head rotates on its hinge. It rotates on its hinge, okay, pushing the actin filament past it. Pushing the actin filament past it, okay. The actin filament moves in this direction. You can see the direction of movement of actin filament, okay. At the end of the power stroke, the myosin head releases and resumes the tightly Riger state, and the same cycle repeats, okay. So this is the sliding filament theory, okay, which is given for the contraction, to explain the contraction of skeletal muscles, given by Huxley and Huxley. The sliding filaments, you can see the band remains same, A band, and they remain same, while the H zone, A and M line remains, A band and M line remains unchanged, and H zone and I band decreases. You can see the sarcomere at rest, and in the contracted state, you can see, yeah, this is the contracted state. Okay, so there is no change in the, A band and Z line. There is a change in H zone and I band. Sarcomere relaxed, contracted. So I have already explained all these steps. Okay, these are the toxins. Okay, which basically can inhibit the process of neuromuscular contraction. Okay, for muscular contraction to occur, what molecules must we have? We know that. Okay, the all the proteinaceous molecules, ATP molecules, there. Okay, rigor mortis is the stiffness of the muscles after death of 72 hours for 72 hours. Okay, so rigor mortis, what is the cause for this? The release of all the calcium from sarcoplasmic reticulum. Okay, it remain in contracted state. We don't have to read this. Yeah, there are some types of contraction. Okay, you have to. So I will be dealing with the types of contraction and the diseases, okay, of muscles in the next turn, and we will be dealing with the smooth muscles and the cardiac muscles. Okay, Cory's cycle. These are the left of the topics. So in the next turn, we will be dealing with all these topics. Till then, bye bye. Take care. Stay safe. Stay healthy.